All right, let's just jump right in. I want you to picture a hijacker. This thing, it's got no brain, no metabolism. It's 1,000 times smaller than a single grain of sand. And it has one and only one goal. Find a host, take over the controls, and force that host to make millions and millions of copies of itself. That right there is a virus. And today, we're cracking open its masterfully simple and yeah, kind of terrifying playbook.
And that's really the big question, isn't it? It's one thing to get sick, but what's actually happening on a microscopic level? How does just one of these tiny invaders manage to turn one of your own cells into a factory that just churns out millions more of them? Well, it turns out they have a surprisingly simple plan.
And this plan, believe it or not, is pretty much universal. You've got countless different viruses out there, but most of them follow the exact same 5-step strategy to get the job done. Think of it like the basic operating system for a successful viral takeover.
So this diagram right here, this is our roadmap. This lays out the entire hostile takeover, from the moment it first makes contact with your cell to the explosive finale where a whole new army of viruses is unleashed. So let's break it down.
Okay, here's the 5-part plan. First up, attachment. The virus has to lock on to a target cell. Then, penetration. It's got to get its instructions inside. Now, step 3 is synthesis, and this is the most critical part, the actual hijacking. The virus forces the cell to drop everything it's doing and start making viral parts instead. Step 4, assembly. All those new parts get built into brand new viruses. And finally, number 5, release. The new viral army bursts out, usually destroying the cell in the process, ready to do it all over again.
So one cell becomes a factory. But what does that really look like over time? I mean, how do scientists even track this kind of population explosion when it's all happening inside a single tiny cell?
This chart here shows it perfectly. It's called a viral growth curve, and it shows something, well, kind of weird. Right after the infection starts, the number of viruses you can actually detect drops to 0. It's like they just vanished. But this isn't a retreat, not at all. It's the quiet before the storm.
This vanishing act has a name. It's called the eclipse phase. So why does this happen? Because for the virus to do its job, it has to take itself apart. It ditches its protective protein coat, the capsid, and releases its genetic code into the cell. For that brief moment, it's no longer a complete infectious particle. It's just a set of instructions, a blueprint, ready to take over the whole operation.
Now, after the hijacking is complete, we hit the maturation phase. The cell's machinery is now a full-blown assembly line, packaging new viral genomes into new protein coats. If you look at the graph, you can see the number of viruses inside the cell just skyrockets. The factory is working overtime, but the doors are still locked, for now.
And when that cell just can't hold anymore, it bursts open. The total number of new virus particles that come pouring out of that one single infected cell, that's called the burst size. And this number can be in the hundreds or even thousands. It's like one person walking into a car factory and a few hours later, 1,000 new cars, each with a copy of that person's instructions, drive out to find more factories.
Okay, so there are so many different viruses out there, right? And they all have their own little tweaks to this playbook. So how on earth do scientists keep track of them all? Well, they needed a good classification system. And that system is called the Baltimore classification. It's honestly brilliant because it sorts viruses based on the most fundamental part of their playbook, what their genetic material is made of, and how they turn that into the instructions for making proteins. It's kind of like sorting computer programs based on their operating system. Windows, Mac, Linux, you get the idea. Just look at this table. You can see the sheer diversity we're talking about. There are 7 different classes based on whether they use DNA or RNA, and if it's single-stranded or double-stranded. It looks a little complex, I know, but don't worry, we're going to zero in on just one of these groups to see how it all works in the real world.
We're going to focus on Group 1, the double-stranded DNA viruses, or dsDNA for short. Their genetic code is structured just like ours is, which gives them a pretty nasty home field advantage once they're inside our cells. This group has some very famous members, like herpes viruses and pox viruses.
So let's put all this theory to the test. Let's take a look at the herpes virus as our case study and see exactly how a Group 1 virus runs its 5-step plan, with its own special twists.
And here it is, the herpes virus, a classic example of a dsDNA virus.
It's responsible for a whole range of common human diseases, and its replication strategy is just a masterclass in biological efficiency.
So let's check out its profile. We know it's got a double-stranded DNA genome. Its protein shell, the capsid, has this cool 20-sided geometric shape. It's also enveloped, which means it steals a piece of the host cell's membrane to wear as a disguise. And get this, between that disguise and its capsid, it has this unique layer called the tegument, which is packed with proteins that are like a special forces team, ready to kickstart the takeover the second it gets inside.
So, right after it gets in, the herpes virus makes a very specific move. Its DNA, which is normally a straight line, enters the cell, sheds its coat, and then immediately forms a circle. Why? This circular shape makes it way more stable and helps it avoid being detected as an invader. From there, it makes a beeline for the cell's command center, the nucleus.
Once it's inside the nucleus, it needs to make copies of that circular DNA, and it uses this incredibly clever and super efficient process to do it. It's called the rolling circle mechanism.
You have to see how this works. It's amazing. Think of a roll of tape. The virus makes a little nick in its circular DNA, and then starts unspooling one strand while it just keeps copying the other one over and over. This creates this super long, continuous ribbon of DNA with tons of copies of the viral genome back to back. Then, it just chops them up into individual units for all the new viruses. It is the ultimate biological assembly line.
And the final steps all happen right there, inside the nucleus. New capsids are built, the freshly copied DNA gets packed inside them, and then, in a final act of pure audacity, the new viruses get their envelope by butting right through the nuclear membrane itself. They literally steal a piece of the cell's command center on their way out the door.
So when you look at this whole playbook, you realize that viruses are the ultimate minimalists. They carry only the bare minimum code they need to hijack these incredibly complex systems for their own very simple goal. And by studying their ruthlessly efficient strategies, you really have to wonder, what else can these tiny hijackers teach us about the fundamental rules of life itself?